recording. So this chapter is about the cell reproduction. And as I mentioned before, in this unit specifically, this chapter and the previous chapter, which is the protein synthesis, DNA and protein synthesis, these are the most important two. It doesn't mean these are the only two, but these are the most important. So pay attention because this is extremely important. Um, and again, I'm going to focus on the core of the chapter, but in this one specifically, it's a lot. But we need to understand it so you can answer any questions. So we are talking about the reproduction. And reproduction, we start with the life cycle, and then how do we reproduce? Um, so reproduction, meaning cells make other cells, basically. So a reproduction can be parent cell, one cell, make new generation, like make two, four, eight, and so on. Example of that is your skin cells. You lose some cells, and then your stem cells, they make two, four, eight, 16, and so on. They make more cells to replace. This is one type. The other type is the opposite. And instead of one making two and four, it is two making one. And this one is two parents produce one. Uh, Most of the time it's one, sometimes it's more, but normally it's one. And this is like the germ cells or sex cells. So like a germ cell from a male, which is the sperm, and from female, which is the egg or the ovum, and they fuse together to make a human being. So these are the two different types, and we will discuss both of them. So touching base on what we already talked about before, and I hope you guys still remember, we usually start with the DNA, the double strand, and here is one strand and the complementary strand, AAT, so this is TTA. You should understand how they fit together, which is basically ATCG. Remember it this way, ATCG. A fit with the T, C fit with the G. So when you see A, you know that the correspondent is T. T, the correspondent is A. C, G, G, C, and so on. This is for the DNA for the double strand. If you were talking about RNA, it's going to be different, and we talked about it. Basically, instead of T, it's going to be U. So those are the two strands, and they are actually attaching to each other by two different means, and I explained that before. One of them is the bases that pair together, like how the A, which is adenine, fits with the T, which is thymine. So they come together. CG, same thing. And the other one is the hydrogen bond. Those are the two things that keep the two strands together. It doesn't stop here because we don't have DNA just like this. It's not loose like this. It's actually coiled around histone. And when it's coiled around histone, histone is a protein. So when it is wrapped around and coiled around protein, specifically called histone, we make chromatin. Chromatin. And in this case, it's not condensed because it's just in the nucleus, not condensed. It's just a normal DNA, and this is how it is located, and this is how it looks like. But when it is time for reproduction, chromatin is going to change to chromosome, which is the same thing, basically. It's just condensed. Chromatin is DNA around protein, but it's loose, not condensed. Chromosome is the same thing. DNA around protein, which is histone, but this one is condensed. Condensed to prepare for the reproduction that we will understand more about it later. The part of the DNA like this, when it's wrapped around, I'm not talking about the whole thing. The whole thing is called chromatin and B chromosome. But this part of it is called nucleosome. Just this part of the DNA around histone. Gene, we talked about that, but still we need to refresh our information to have better understanding. On the DNA, something like this, again, each piece of it, like this part right here, this is a gene, another gene, another gene, another gene, and so on. So DNA contain a lot of gene pieces on the DNA called gene. And the DNA will have a lot of these gene. The DNA is very, very long. So every patch of the DNA is called gene after gene after gene after gene. How many genes? We don't have to know the numbers, just know that it's a lot, and different chromosomes have different numbers without going into the details. This is good enough to know. And the other thing is this chromosome has a specific number of genes, and the other chromosome can have a different number and also different type. So each chromosome will have different number, different type than the other ones. And one more thing, the gene, I said it's a piece of the DNA. It's this part of the DNA that code for a specific protein. This is the remaining of the definition. It codes for a specific protein. So what's a gene? It's a piece, a segment of the DNA. DNA have a lot of it that code for a specific one protein. We call that a gene. Now, how many chromosomes do we have in our cells? We have this set of chromosomes, which is 23 pairs. So the total number is 46, but it's not scattered. It is arranged in pairs. What you see here is something called karyotype, and you need to understand that. Karyotype means the specific arrangement of the chromosomes when we have like we use certain dye, like we take it out of the nucleus, they spread it in the laboratory, and they add fluorescent dye to it to highlight these chromosomes. And when you look at it under the microscope, this is what you see. So we call that karyotyping. Karyotype or karyotyping means show me how exactly it's arranged, the 23 pairs, how exactly they are arranged and how they look like. And if you noticed, they are arranged to a big extent according to their size. Look at this and this. They look like each other. Not perfect, but it's very close. This looks like this. So it's not like one of those will come with one of those. No, it doesn't happen like these. These are two similar chromosomes, and that's why they come after each other. And they will call it like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Stop. Stop at 22. The last one, which is 23, is different than the other 22. From 1 to 22, this is in all cells, males or females. The 23rd, also in males and females, but this one is different. This is the 6th chromosome, which is pair number 23. So if you look at this, how many chromosomes do we have? 23 pairs, which is 46 total. You should remember that. You can't say 23. No, we don't have 23. We have 46. We say we have 23 pairs of chromosomes. Since it is pairs, we call that diploid. And dipole is a prefix for double. It's the Greek or Latin for double, just to remember it. So dipole, double. It's double because it is duplicate of each one of those. Like this is 1, 1, 2, 2, 3, 3. So we call that diploid. You have 2 of each of those. So you end up having 23 pairs pairs, not singles. If you have 23 singles, we don't call it diploid. You will see the other name. It's called haploid, hapl. So dipl and hapl. Remember these prefixes. Dipl, double. Hapl means half the double, which mean single. Dipl, hapl. Diploid, haploid. And this is exactly how we count. We count from 1, and this is 23. 23 is the 6th chromosome. Each two of those that come together, like here, 1 and 1, 2 and 2. Those together are called homologous. Homo means similar. Homologous. So this is 1 and this is homologous 1. So those two are homologous to each other. We call that homologous chromosomes. That's why they were paired together. And they are homologous, kind of similar, but one of them is coming from the mom and one of them is coming from the dad. One of them coming from the mom means when it was still an egg. This is what the egg is giving you. Like, let's say this is chromosome number 1. This one, single one coming from the egg, from the mom, and this one that will pair with it, that will come with it, that's homologous. It's coming from the dad, meaning from the sperm. And we call those together diploid or a pair. So it's not random. It's one and the other one is homologous. It's very similar to it, but one from the mom and one from the dad. So all what we talked about so far is diploid, which is, we call this is in somatic cells. All what we talked about so far, the 46, the 23 the 23 pair from 1 to 23, the last one is the 6th chromosome and all of that. All what we talked about so far and what you're looking at right here, this is in all somatic cells. What's somatic cells? Somatic, what does it mean? Somatic is coming from soma, which means body. So skin cells, yes, 23 pairs, just like this. Liver cells, yes. Bone cells, yes. Any cell in your body, yes. All of them have the same karyotype that you saw. The only exception to that is the gametes or the sex cells, which is sperm or ovum. They look different. It is completely different. This is the only one type of cell that is not 23 pairs, just like any other cell. It goes without saying. You name it. Any cell in your body is autosome, which is somatic. It's somatic. Somatic. Soma, body cell. Body cell like what? Anyone. Skin, bone, stomach, blood, anything, nerve, anything. All of them contain 23 pairs. The only one exception is gametes or sex cells. So those have 23 singles, not 23 pairs. So the total number is actually 23 because they have 23 singles, not like this. This is 23 pairs. The total is 46. This is 23 singles or haplo. Look at this word here, haploid. Haploid, we call it, it's one N, like one set of chromosome, 23. The other one, we called it diploid, 2 sets. So we call it 2N, like here. The diploid is 2N. What 2N? 2 sets, which is like 23 pairs. Here it's one set, 23 singles. And we call that haploid. What's haploid? Hapl, as I mentioned, is half. Hapl, half. Dipl, double. Hapl is half of the double, so it is singles. So the sperm will have 23 of those, and the ovum will have 23 of another 23 of those, the corresponding 23. There are other things beside haploid and diploid. There is triploid, and obviously hapl, double, triple, quadruple, or tetraple. So it's basically it's Greek and Latin for one, two, three, four, and more, and multiple. So happle, single, dipple, double, triple, three, tetra, four. Poly is anything more than two. So triple or tetraple or even hexaple and heptaple and octaple. All of these are numbers that correspond to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Instead of that, they say happle, double, triple, tetra, um, penta means five, tetra means four, hexa means six, hepta means seven, octa eight, deca is nine, and so on. So this is not our area of interest. Just know that in other, not humans, we're focusing. This is human biology, so we're focusing on what we actually have, which is haploid and diploid only. But just understand that in other creatures, in other like plants and animals and fish and other things, they have different things. This is the bottom line. So it can be like triple, like three, like this here. They have three of each type of chromosome, like in salmon, in banana, and stuff like that. Poly, which is whatever the number that's more than two. It can be in like strawberry, have seven, and so on. You don't have to pay much attention to that. It's more important to know happle and dipple, which is in humans. So out of the 23 pairs that I talked about, from 1 to 2, we call it autosomes, which is somatic cells. Auto. Auto is same as somatic, auto. So autosomes are the somatic, somes means somatics. So these are the somatic cells. Somatic cells. If you get one of your skin cells, if we like go in the laboratory and open the cells, they have the tools to do that and open the nucleus and get the chromosomes out. They will find that from 1 to 22, these are just the normal or regular chromosomes called autosomes. The last one, which is the sex chromosome, which is number 23, which is called allosome. This is the one that would determine the biological sex. If both of them are X's, X and X, this is female. If it is X and Y, it is a male. So look at this. From 1 all the way to 22, these just are called autosomal cells. Understand that. This is important. This is part of the chapter that you need to know. So from 1 to 22, what are these? Autosomal cells, autosomes. Autosomal chromosomes, I'm sorry. Autosomes or autosomal chromosomes from 1 to 22. Males and females, no much difference. No much difference. But the last one is different from males and females. So if I get skin cell from me and I look under the microscope, I will see 23, but number 23 is XY. If you are a female, do the same thing. I will look at 23, you will see XX. And I'm talking about normal now, because there are some abnormalities. We will just briefly talk about it, but I want you to focus more on what's normal. So number 23 is not an autosome. It is a sex chromosome. It is an allosome. It's different from male to female. Here is a male. Look at this. Number 23. From 1 to 22 is not different. Males and females, it doesn't matter. It's the same thing almost. It's very similar. But look at the male, it's X, and the pair, the homologous pair is actually Y. And look how Y is very short and tiny. This is Y. If it was an X, it will look like this. So if this is a female, you will see another one looks exactly like this one. If you look at this karyotype, you can tell right away that this is a male immediately, because see how this is big and this is how it is like very small compared to the X. If you see like this and another one that the same length, Beside it, this will be XX, and this is female. So remember that the Y is tiny, the X is big. I'm talking about number 23 specifically. So this is all what I talked about so far. This is normal. Here are some abnormalities. Just take a look at it. I'm not going to ask you like to memorize all these abnormalities, but just look at this Klinefelter syndrome. Look at number 23. And instead of having XX or XY, it has two X's and one Y. So it's trisomy, trisomy. Instead of having two homologous chromosomes, two or just one pair, two chromosomes, which is one pair, and number 23 can be either XX if it's female, XY if it is male. No, here we have XXY. Is that a male or female? It's a syndrome, which is abnormality. Uh, you can't say it's female because there is Y. You can't say it's male because it's X. But since you have Y, it's tending more toward males because females basically do not have Y. If you have Y, it will give this human or this person the characteristics that come with the Y. So it's more of this is not accurate. I'm just going to give you a description that just help a little bit. It's like a feminized version of a male, something like this. This is not accurate. It's you have XY, but you have an additional X, and we call that Klinefelter. And this is an issue with number 23 specifically. Another issue, but this one is not in the sixth chromosome. The abnormality here is in pair number 21. And this one is one of the autosomes. Same thing, instead of having two, which we call it pair, there is an extra one. So we call it trisomy. Look at this, tri. Tri means three. The very famous Down syndrome. I don't know if you guys heard about it or not, but it's very famous. It's trisomy. This is trisomy also, but trisomy in the sixth chromosome. You have three, three. Here, the trisomy is in one of the autosomes, specifically 21. There are other forms. I'm not going to ask you to know the details of Jacob and Turner and all that stuff, unless if you are interested to know that, that's okay. But otherwise, you don't have to. If you're interested only, I can give you an example here. This one here, like XXX, you're supposed to have two X's only. You have an additional X. Occasionally, they call it a super female, like very female. This is like they call it the super male because you do have XY, you are a male, but you have an additional X. This one, O here means zero, like this, but they put an O instead of saying zero. So what is this? It is less female. It's a female. Why did you say female? Because she doesn't have an X, a Y, I'm sorry. She doesn't have a Y. Once you don't have a Y, I have to call you a female. So Y, it's all about Y. If you have Y, I will call you a male. You don't have a Y, I call you a female. You should have two X's. What if you have one X only? Yes, we call it Turner syndrome or XY. And if that's the case, it's a female, but she's short and missing a lot of things. We don't have to go into the details. If you're interested more, you can read about it, but you're not responsible for that. So we mentioned that before. You should be able to answer easily. DNA is organized into chromosomes. The two types of chromosomes are, this one here is autosome, and the other one is sex chromosome. This is important. You should be able to tell. Autosomes are chromosomes other than 6 chromosomes, and the 22 are called autosomes. Allosome, this is number 23, which is the 6th chromosome. Human cells, we're referring to the somatic cells, the body cells. the normal cells, not the sick cells. Contain 2N, which means two sets. So it's two sets of 23. The total number will be 46, obviously. And one set is called haploid, and the two sets are called diploid. Haplo, dipple. Haplo and dipple. Half or single and double. Haplo, dipple. Single, double. One chromosome from each of the 23 come from the mom and one from the dad. And the last one, the gametes are also called what? We call it 6 chromosomes. Which is half or haploid, which is this. It's called haploid. And 6 chromosomes can be XX, and this is what? Or XY, and this is female, and this is male, obviously. Okay, now the next part is very, very important. This is from the core of the chapter. So expect to see a lot of questions here. Okay, so be ready. And let's talk about how the cells reproduce. This is extremely important. Lecture and lab. Okay, so pay attention. How do we reproduce or divide? Well, this question, to answer this question, I have to ask you another question. Which cells are you talking about? Like you said, how do we divide? In the first slide, we said reproduction can be from the body cells, autosomes, the body cells, any cell in the body, 1 give you 2, 4, 8, and so on. The opposite is 2 give you 1. So 1 give you 2, give you 4, 8, 16, 32, and so on. This is in the somatic cells. Somatic cells, body cells, all cells, you name it, except germ or sick cells. We also call it gametes. And this type of division is called mitosis. To understand, mitosis is coming from mitos, and mitos means thread. You will see that there are actually threads that attach to the chromosomes, and when they pull them apart, they are going to divide. And this is just if you wanted to understand this. So mitos is a Greek for threads. Threads attach and pull away. And again, you lose your skin cells, you have to replace it. So that's why your stem cells, 1 will make 2, will make 4, 8, 16, 32, and so on. So you lost 500 cells, you're going to make another 500 cells this way, which is called mitosis. The other thing is called meiosis, and meios is a Greek for reduction. I highly advise you guys to collect and understand these prefixes and suffixes. It's going to help you a lot. So what's meios? Meios means reduction. So because you start with 2 and you end up having 1. You start with 2 sperm and ova. They merge together and give you 1 cell, which is a fertilized egg. So 2 give you 1. 2 sets give you 2 sets of chromosomes. I'm sorry, 2 sets give you 1 set. So this is called meiosis, and this only happen in the germ cells. And we call that the sexual reproduction. Germ cells, gametes, sick cells, same thing. Germ cells, gametes, sick cells. We're referring to the sperms and the ovum. So mitosis, very important. Lecture and lab. Life cycle. Let's start with the life cycle, including the mitosis. So the life cycle, and remember, mitosis alone is, we're talking about somatic cells, any cell in your body, but not the germ cells. So there is a cell cycle, life cycle for each cell. Like red blood cells, for example, they live for 120 days, like 4 months. Other cells, epithelial cells in your stomach, they stay like for a day. So you replace them like every 24, 25 hours. Bone marrow is even shorter. Its lifespan stay only for 18 days. Red blood cells, 120 days. They stay for a long time. Other cells, some of them, we don't even reproduce it. Um, like the nerve cells and the muscle cells. We will discuss it. But here is the different phases of every cell of the somatic cells that we are talking about. Remember, I'm talking about mitosis now. Mitose, threading. One, give you two, give you four. So we're referring to what? Somatic cells, not the sick cells. Sick cells, meiosis, this is another topic. We will discuss it later. But for now, I'm talking about mitosis, somatic cells. 1, 2, 4, 8, 16, and so on. And this cycle is divided into interphase, and then mitosis, and cytokinesis. Interphase is the longest of all. And enter, another prefix, it means in between. In between what? In between mitosis and cytokinesis. Like here is mitosis, cytokinesis, and then you go interphase, 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 until you come back to mitosis and cytokinesis. And after you do it, you go back to interphase, 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 and so on. So it's between mitosis to the other mitosis. It's in between those two. Mitosis, cytokinesis, and then interphase, in between each two successive mitosis and cytokinesis. So mitosis, cytokinesis, interphase, in between this mitosis, cytokinesis, and the next one. And that's why we call it interphase. Interphase is divided into 3 steps, and mitosis is divided into 4 steps. We have to discuss the details. Interphase G1, and G is coming from growth. This is growth number 1, when the cells start to grow, grow, grow. Preparing the cells for the next, which is mitosis. And so this is for the different parts of the cells, but not for the chromosomes. Here are the chromosomes, S. And the S is the first letter of synthesis. They are referring to DNA synthesis. You have 23 pairs of chromosomes. You're going to duplicate. You will have two sets, duplicating. Instead of one set of 23 pairs, which is 46, you will have 2 sets. Each one is 23 pairs. Remember, this is not reduction. It's diploid. Diploid. This one cell is 23 pairs of chromosomes. You have to duplicate that. So when you divide, you end up having 2 cells identical to the first one. Why? Because you don't want to have abnormal cells. You're having skin cells. What do you want to produce? 2 skin cells, just identical to the first one. From those 2, you get 4, exactly the same, identical. Why do you want to make different skin cells than the one that you originally have? You don't want to. So it is identical. In order to make it identical, like I have a copy of the slide, I make another copy of it. So now I have 2 copies, exactly the same. This one you take it, and this one I take it. So there is no reduction. It was 1, I make it 2, and I give you 1, I take 1. So you duplicate first. Duplicate what? Everything, but the S is specifically for DNA duplication. And then the G2, just like G1, but you do more in order to prepare the cells for mitosis. Mitosis will go into 4 phases, and the 4 are P-mate, P-M-A-T. This is an acronym to remember it. P-mate. P is for prophase. Pro, phase. Phase, phase. Stage, phase. Pro means beginning. Pro. Metaphase. Meta means equator or middle line. Meta, middle line. Metaphase. Anaphase, the phase of separation. Ana means apart. I'm giving you the exact translation, by the way. If you look it up, you will find this is the translation from Greek and Latin to English. Okay? So ana means apart. So instead of being like this, apart, you see, you start to separate. Telo means distance or away. Like if you think about it, why did we call the telephone? Why did you call it telephone? Phone is sound. Telo means away, away. Like you hold it, your telephone, and somebody else in another state is talking to you, and the sound is moving away or in a distance. So telo means distance. So this is the distant phase, and you will see the details. I just wanted to translate it for you so you have an understanding of what does it mean first, and then we go into the details. The last one here is cytokinesis, and we will cut it also into two pieces. Cyto means cells, kinesis is moving. So you now you have two cells and they move away. Cytokinesis. The daughter cells move away. So let's start with some details about the interface. The interface is the phase here. This is mitosis right here. Prophase, metaphase, anaphase, telophase, all the brown. So from here, you start your interface between this mitosis, interface, 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 until the next mitosis. You do the mitosis and enter back into the interface until the next mitosis, and so on. So the interface right here is divided into three parts: G1, S, G2. G1, the cell grow. Grow for what? Just normal growth, and also to prepare and provide what the cells need to duplicate the chromosomes, preparing for the S. S is the actual duplication. S synthesis, DNA synthesis, the actual duplication. And then G2, this is growth again. G is growth, but this one is more specifically proteins, and including also other things that are going to grow so that you enter into mitosis. This is the interface. Now mitosis, P mate, prophase, metaphase, anaphase, telophase. These four. That would be followed by cytokinesis. Here it is again, G1. G is the first letter of growth. So this is growth number one. Growth of what? Factors needed to duplicate, which is S, to duplicate and synthesize and duplicate DNA. S is the synthesis, which is when you duplicate, synthesize, and duplicate the DNA. And G2 is DNA is already too duplicated already. So you're going to make the protein needed for the mitosis, the factors that are needed to repair DNA. If there is something wrong, you do it in G2. You provide the threads that are going to help you with the mitosis, with the division. So you prepare and repair, okay, until you go to the M, which is mitosis. It's important here to understand as well. Look at these check marks here. Here is one, here is one, here is one. After you finish each phase, there is a check mark that can stop you or let you go. And this is very important for us. So you're working on G1 here. So you're making the factors that are needed for the synthesis protein, for the chromosomal duplication or synthesis of the DNA. you're working on it and you're preparing, and then we will tell you, stop here. Is everything good? Did you make sure that you have all what you need? Is everything in good shape? Yes. Okay, move on to the census, to the S, and then move on to the G2, and then stop. Now, listen, we are going to mitosis. Is everything ready for mitosis? Did the chromosomes duplicate? Yes. Did you repair any damage? Yes. Did you make everything ready? Threads, centrioles, and other things needed? If yes, then go ahead and move to mitosis, prophase, metaphase, anaphase, telophase, and then stop. I'm going to the G1. No, stop. We have to make sure that these are all good. Cytokinesis is happening, and then I will allow you to go to G1. And one more information that's important here that I want you to know is the importance of that is not only to make sure that everything is ready for the next one. This is very protective against cancer, tumors. If we don't have these check marks, the cells will go like nonstop. G1S, G2M, G1S, G2M, and go like cycle, 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 very fast. So we end up doing unlimited number of cells, which is tumor. But if you start to go fast, the check marks are going to stop you. So do you mean that like these check marks can be removed for people having tumor? Yes, there can be an issue. This is not the only reason, but the reason here. This is one of the reasons of creation or formation of tumors. The check marks are not good. So they don't stop you. If you go fast, they don't stop you. There are other reasons. We will talk about it later. And we're not going into the depth. I'm just giving you a general idea. So now details of mitosis. Mitosis, does it happen in somatic cells or germ sex cells, also known as gametes? Somatic cells. What do you mean by somatic cells? Body cells, any cells, skin, liver, bone, anything. So you start with one cell, you make two identical cells, 4, 8, and so on. So what are the steps here? You go from here, prophase, early metaphase, and here's the metaphase. Meta means equator. Pro means preparation. Metaphase, middle. Look how the chromosomes are in the middle, equator. And then the anaphase, look how they are moving apart, ana, apart. Another one. And then when they move away from each other, this is called the telophase. Look how this is in one direction, in one side, and this is in another side. So it's telo, away. Cytokinesis, they move away from each other and they become two cells. And look at this. This one cell will also divide. This one cell will divide. So in the next cycle, it become 4, 8, 16, and so on. This is mitosis. Let's go through the details. Prophase, metaphase, anaphase, telophase. P-mate. And then cytokinesis. So here, just making sure that you got what we talked about. The cell cycle is the life of somatic or sex cells. What do you guys think? When we say the cell cycle, like all these steps, G1, S, G2, mitosis, all of this is a cell cycle. Are we referring to the somatic cells or the sex cells? Does anybody know?
Somatic.
I'm sorry?
Somatic.
Yes, somatic cells. Sex cells is completely different. It's meiosis. The cell spends most of its life in which one of those?
Interphase.
Interphase, yes, very good. The cell cycle has two main stages. The first one is The two main stages. Interphase, and the other one is
Mitosis.
mitosis. Yes. Interphase is divided into three: G1, S, G2. G1 is growth number one. S, what here is copied and duplicated?
DNA.
DNA, and chromosomes are duplicated. G2 is preparing for what's going to be next here?
Mitosis.
Mitosis. Which points here would delay and it actually can stop moving from one to one?
Checkpoints.
Checkpoints, yes. And I mentioned checkpoints for the tumors. If you don't have the checkpoints, if somebody have an issue, a problem with the checkpoints, they can develop tumor because they delay and they stop you. Mitosis is four parts, which is the P mate. P is
Prophase,
prophase, M,
metaphase, anaphase, and telophase.
excellent, excellent, yes. And what is this when the cells actually divide and make two? They move away from each other, make two identical cells.
Cytokinesis.
Cytokinesis, yes. Okay, so let's get into more details. First of all, as far as I know, you guys need to do that in the laboratory as well. You do it somehow. Even if it's virtual, you will need to see the different steps. So let's understand first that we have something called centrosome, and we talked about that in the cells in the beginning, if you remember. What's the centrosome? Centrosome is microtubules organizing center, a center that organize microtubules. Okay, and it's made up of two centrioles. Look at these two green things. Those two are called centrioles. Together, it's a centrosome. Centrioles, two of them. And look how one is like this and the other one is like perpendicular to it or 90-degree on it or right angle on it. Yes, this is how the two are arranged. Two what? Two centrioles. What do you call them in this arrangement? Centrosomes. We will understand why do we have to know this, because this is the one that's going to make the threads and help you to divide. So if you look at this chromosome, let me ask you this. Is a chromosome DNA with protein that is condensed or loose? I'm telling you that chromosome is DNA around protein called histone. Is it loose or condensed? Loose, which is uncondensed, or condensed? Does anybody know? The question is condensed or not? Anybody know? No? Okay, this is condensed because we're preparing for division. If it is not condensed, we call it chromatin, not chromosomes. So it is condensed. Why is it condensed? It was chromatin, loose, uncondensed. Why did you condense like this? To prepare for division. So here is a chromosome, and it will duplicate. It will make two during the S phase, duplication. So one chromosome become two, but the two are not separated yet. Here is one that's called chromatid, and the other one is the sister chromatid. Why did you call it chromatid? Because it's not a full chromosome yet. It's not ready yet. It's just a chromatid, and when they separate and it will become a complete chromosome, we call it a chromosome. So chromosome It's the DNA with the genes around protein called histone, and these are the sister chromatid or chromosome, and it's identical copy, but not separated. You make a copy, they did not separate yet. And they are attached. The point of attachment is called centromere. Centrosome is what I talked about, is the centrioles that are right angle to each other. And this is what's going to make the threads or the spindles in order to divide. So look at the first one, prophase. Remember P-mate, P-M-A-T, then cytokinesis. P-mate, cytokinesis. P-mate, which is mitosis, P-M-A-T. Prophase, metaphase, anaphase, telophase. Prophase, this is the first one. Prophase, there is an early part of it and late part of it. Early part, what happened here? And you can be asked about the details here. You need to know what happened. In the prophase, early prophase, what happened? Number one, the centrosomes that we talked about, one of them will come into one pool, the other one come into the other pool, and they make spindles extending between them. What are the spindles? Microtubules. So one centrosome come here, the other centrosome come here, and they make spindles from the microtubules extending between them. And you will understand why they are doing like this. This is one. The other one. Look at this. See, it should be all purple, a complete double wall of the nucleus. This wall, I'm sorry, not a wall, a membrane. This membrane is going to start to disappear. So look at this, disappear, and then the rest will disappear. You end up having no membrane. The whole membrane will end up disappearing later. But this is starts to disappear. This is one, or actually two. This is one, nuclear membrane start to disappear. And the third one is the DNA change from chromatin to chromosome, meaning it condense. And it make the two sister chromatid. The duplication is already there, but you have two sister chromatids. And then in the late prophase, still prophase, but whatever remains from the nuclear envelope or membrane will go away. And these centrosomes that we talked about will start to attach to the chromosomes. They start to attach the chromosomes. They attach to the chromosomes in the center here. So this is prophase. This is where we make the spindles from the microtubules. We mentioned already centrosomes go to the pools, the nuclear envelope disappear, start disappearing early, complete the disappearance in the late. The DNA condense, change from chromatin to chromosome. It's two identical chromatid held by centromere. The center that's holding them is called centromere. Spindles start to attach from the centrosomes to the chromatid, specifically attached to the centromere. Next one is metaphase. After they attach to the chromosomes like this, they are going to arrange it in the middle line. And this is called metaphase. Translation, what's a metaphase? Meta means equator. Look at this arranged, or the middle line. Here is the equator here. They arrange around the equator right here. This is the most important point for the metaphase. So here it is. You arrange in the middle line, like you make a straight line like this. And the two sister chromatids are, as you see here. One is attached to this side, and the other one is attached to the other side. Attached from what? From the centromere. So it attached these spindles, the microtubules, attached to the chromosomes. Which part specifically? Centromeres, where the two copies of the two sister chromatids attached to each other. They attach here. So this is metaphase, which is a line along the equator or the middle line, while the spindles attached to them. Anaphase. Anaphase, I told you, means apart. Ana, apart or separation. Look at these spindles. They will start to contract. Each one of them is attaching from the centromere, which is the middle part, and they will pull it apart. See how they are pulling away? This is called anaphase, ana, apart, which means separate. So this one is having one of the sister chromatids, and this one is having the other one. They are not connected anymore. They are not connected through the centromere anymore. One of them pulls in this way, and the other one is pulls in this way. So it's just separating. They didn't move apart. It's not like they didn't move like far away from each other. They just separate. And instead of being together like this, they just separate. Separation is anaphase. Here it is. Anaphase, the spindle fibers contract, pulling the sister chromatid away from each other. Telophase, I told you telo, like telephone, means distance. So now, completing pulling away of these sister chromatid, so each set of those will be in one side, and here is the other one, and us in the other side, and they are away from each other. We call that telophase. Telophase, away. A group is here, and the other group is completely separated, far away in the other side. So they are telophase now. They are far from each other. Not only that, the other thing that happened in the telophase, but telo is coming from this. Is that the only thing that happened? Like this set is here, and this set is far away in the other side? No, that's not it. Other things will happen. What are the other things? The nuclear envelope that we said it disappeared in the prophase start to reappear in the telophase. Look at this. Start to reappear. The other thing is there is something here in the middle that's called cleavage furrow. Cleavage is coming from cleave. When you cleave means you cut away, separation. Furrow means this, depression here. So this is demarcating this part right here in order to separate. But you almost have a cell here and another cell here, but they are not separated yet. There is just a cleavage furrow in between. So here it is. This is what happened in telophase. One more thing that I didn't mention, which is chromosomes that was condensed, they become uncondensed now, become chromatin. Last one, look at this cleavage furrow right here. It's going to go deeper, 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 until you have actually two cells that move away from each side, and we call that cytokinesis, which follow the mitosis with the four stages, PMAT, prophase, metaphase, anaphase, telophase. We talked about those with the details. Now, when the actually two cells move away from each other, it is one daughter cell, it is another daughter cell. This is called cytokinesis here. And each one of these cells contain everything. Look at the nuclear envelope. It's complete. Chromosomes have a complete set here. This is the centrosome here. This is the same thing exactly here. Two identical daughter cells. Each one of those is going to undergo the same thing again, but not right now. They are going to go through the whole cycle again. There is a video here. You can watch it. You might watch it in the video. I don't know how they're going to do it, but this is summarizing what we talked about. Here it is. There is a G1, S, G2, and then the prophase, look at how these aster or the centrosomes, one here and one here, and in between the mitotic spindles or the microtubules, and look how the envelope here start to disappear. Here it's disappearing even more, and the spindles are attaching, which is the pro-metaphase, and then they arrange around the equator, which is the actual metaphase. And then they separate from each other by pulling of the spindles, anaphase, cleavage furrow, reforming the nucleus and the nucleolus, and start the separation. And then when it move away, we call that cytokinesis. And this is a question about what we talked about. What is microtubules organizing center? The name give it to you, center. So it's centrosome. Centrosome, microtubules organizing center, which is in the cytoplasm. And it contains centrioles, which is two right angle. What are the two cylinders that I just talked about? Right angle, centrioles. What or how DNA organized? Chromosomes. We talked about that several times, chromosomes. So if I ask you how the DNA is organized around the proteins, chromosomes. DNA is not loose. It's not like scattered. No, it is organized around protein, and we call that chromosome. What are the two identical sisters in the chromosome? We call that chromatid. And last one is centromere, is where those two chromatid attach to each other before the separation, before the anaphase. So this is, again, summarizing what we already talked about. Prophase, metaphase, anaphase, telophase, and cytokinesis. Okay. Next part is going to be the 6 cells or the germ cells, and we will discuss that in the other lecture, in another meeting. So we finished somatic cells with the details about the karyotype, the chromosomes, and how the cells divide in mitosis. Which cells? Somatic cells. Next time, we will talk about how the 6 cells work and how they fertilize, fertilization and the rest of it, which is meiosis. So that will be in part 2. So I'm going to stop here.